This is GATE 2022 Life Sciences, Section P Biochemistry. Question number 30. Which one of the following statements about DNA polymerase 3 of E. coli is not correct? A. It catalyzes Nick translation. B. Its absence is lethal to E. coli. C. It synthesizes a complementary DNA strand using single-stranded template. D. It possesses 3' to 5' exonuclease activity. This is the NPTEL online course reference, okay, which explain the basic biochemistry of different polymerase enzymes. So here we are talking about DNA pol 3, which is found in prokaryotes, mainly E. coli. So E. coli has 5 DNA polymerase. Okay, and what is the main role of this DNA polymerase? It polymerizes the DNA, right? Simply. And chemically, as you can see, it is a DNA dependent DNA polymerase. And by that, what I mean DNA dependent, that is, the template will be DNA itself, and it will add different units of NTPs. It will polymerize the complementary strand on this template, okay, and this always polymerizes by in a 5' 3' direction. Okay, the polymerization is always in 5' to 3' direction. So at 5' direction we have a phosphate group, and at 3' we have a free OH group. So from this free OH group we have a incoming nucleotide triphosphate. Okay, this is NTP, nucleotide triphosphate. It will attack on this 3' OH and it will form a phosphodiester bond. Okay, so when the phosphodiester bond is formed, it releases a diphosphate. Okay, so during this step the diphosphate is released, which is converted to pyrophosphate, and this conversion from diphosphate to pyrophosphate releases an amount of energy which causes the formation, which helps in bond formation essentially. Okay, so this is the simple biochemistry, like the chemical reactions which takes place. It is a very important DNA enzyme, which is DNA polymerase, which polymerizes our DNA. Right, we have DNA replication from this enzyme. So in E. coli, as I mentioned, there are various different DNA polymerases. So in E. coli there are 5. Okay, so DNA polymerase 1, which is coded by Pol A gene, is a major repair enzyme. Okay, this is also very important DNA polymerase, both in E. coli as well as it has use in various molecular biology technique. We will discuss this also. Then we have Pol 2, DNA Pol 2, which is a replication restart. So it is generally it is generally involved in during the starting of the polymerization reaction. It adds few bases during the start of replication. Pol 3 is a replicase. Pol 3 is a major DNA polymerase. It will polymerize mostly the long strands of E. coli. So major DNA polymerase from 1, 2, 3, 4, 5 is Pol 3. Okay, it's a major replicase enzyme. It will mostly polymerize the DNA fully, and these all are repair and helps in replication start. DNA Pol 4 and 5 are the repair enzymes, and these are mainly specific, which carry out trans-lesion replication. Okay, this is also very important. Trans-lesion replications, these are found during, these are the repairs which occur during replication, and these enzyme recognizes this error during the replication and they repair it. So all these enzymes, the one common thing in all these enzymes, they have a 3 prime to 5 prime exonuclease activity. Okay, what I mean by that? So exonuclease means it will so there are two types of nucleases. Nucleases are those enzymes which cleave the DNA, right? They cleave the DNA. So nucleases can be of two types, either exonuclease or endonuclease. So exonuclease will cleave the DNA from the outer edges, okay, whereas endonuclease can cleave from in between also. They can cleave from this phosphodiester bond or this, whereas exonuclease will always target the ends of the DNA. Okay, so all these DNA polymerases, they have a 3 prime to 5 prime exonuclease activity. And by what I mean by this exonuclease activity is, so the replicase enzyme, for example DNA pol 3, it is adding, okay, it is adding adding each base pair. Okay, suppose this base pair which they have added is wrong. Okay, the DNA pol 3 has a recognition domain, a subunit on it, which recognizes that this is a wrong base pair. So what it will do, it will remove this base pair. So this removing of this base pair has a direction of 3 prime to 5 prime. Okay, so this exonuclease activity is a repair activity of all these polymerases, that they'll recognize the mismatch or the wrong base and they'll remove this from 3 prime to 5 prime. Okay, they polymerize from 5 to 3 prime, and this exonuclease will remove from 3 to 5, right? So all the polymerases, they have a basic 3 prime to 5 prime exonuclease activity. This DNA polymerase 1 is a special type of polymerase. It has a 5 prime to 3 prime exonuclease activity. And what is this exonuclease activity? So this can be explained using Nick translation. This process is known as Nick translation. So what happens? Suppose there is a duplex strand, okay, DNA duplex strand, and we have a nick, right? We have a nick in between. So this nick in between is observed during various this nick is observed in various cases, like suppose there was a primer here, and now the primer is replaced, and this primer is replaced, and there are bases are added in place of that primer, and this primer was still here. So someone has to come and has to ligate this nick. Okay, we have ligases also. Suppose this nick is not ligated, okay, or we have a nick generated here because of exonuclease activity also. So what happens in DNA, the DNA pol 1, it will sit on this nick, right? And as I said, this has a special activity known as 5 prime to 3 prime exonuclease activity. So it will sit on this nick and it will degrade the old strand, right? It will degrade the old strand. So this blue strand, it will degrade completely, and now the nick is no longer here, and it will replace this blue strand with this red, which is matching correctly, okay, which is matching correctly to the template. It is just replacing the whole strand so that this nick is going to be replaced, and this process is known as nick translation. Okay, this is very important. How it is very important? So this nick translation was first found in the DNA pol 1. It was then further used in mol bio techniques. So what we can do, we can replace we can synthesize a newly new strand on this, on the pre-existing template, and this bases could be either radioactively labeled or they could be fluorescently labeled. And now thus we can label our duplex DNA. Okay, we just need to have a nick and we can label a duplex DNA in our lab, and then we can use it for various application. Okay, so this is the application of nick translation. Okay, so coming back to the question, which one of the following statement of DNA pol 3 of E. coli is not correct? So as I mentioned earlier, it does not catalyze nick translation. Nick translation is catalyzed solely by DNA pol 1, right? So the second option is its absence is lethal to E. coli. Obviously DNA enzyme, as I mentioned here, it's a major replicative enzyme. If you remove this pol C gene from E. coli, the E. coli won't survive because it's a major replicating enzyme of E. coli. So its absence is lethal to E. coli. This is also true. It synthesizes a complementary DNA strand using single-stranded template. This is what all polymerases do. They synthesize a complementary DNA strand using a single-stranded template, right? DNA-dependent DNA polymerase activity of DNA polymerase enzyme. It possesses 3' to 5' exonuclease activity. Again, this 3' to 5' exonuclease activity is present in all the polymerase, right? As I mentioned here, if any polymerase observes any mismatch or a faulty base, it will go back. It will remove that faulty base. It will remove more bases just to be sure, and then again it will restart its journey by polymerizing the strand. So the going back in the opposite direction is known as 3' to 5' exonuclease activity, and yes, it is present in DNA pol 3. So the correct option is A. DNA pol 3 doesn't catalyze nick translation, right? DNA pol 1 catalyzes nick translation. So the correct option is A.